Right, hi guys. Wanted to make a short video on exam technique here. Basically because I think like as teachers, we might be guilty quite a lot of talking about what exam technique is and how it works, and maybe be a bit general and vague to students and not maybe highlight what that means. So what we're talking about exam technique is when you actually answer a paper question, are you able to apply your subject knowledge from the specification to the question and interpret what the question is asking to gain maximum reward for that in terms of the marks? So first thing I'm doing whenever I look at a question is I look to see how many marks are available. So this one is worth 3 marks, so I know I'm looking there to make 3 clear points. The second thing I'm looking at when I read the question is, as I go through the information here at the top, we're looking at antibody structure. This is taken from an OCR A-level biology paper. And what we're doing is we're looking to see the information there, but just make sure that we fully understand the stem of the question. That means all the background information we're given. We understand the stem of the question so we're able to answer the directed question at the bottom. So it says, an antibody is an example of a protein molecule. As I'm going through, I'm underlining the key terms that I might need to know about here. So I see an antibody is an example of a protein molecule, and it says it has a specific 3-dimensional shape. So as I'm going through that, I'm possibly kind of thinking, all right, so in the book, or from the lessons I've been in, I know that 3D shape means tertiary structure. And then it says the diagram below shows the structure of an antibody molecule, and I've got the constant region here and I've got the variable region above. I can also see light polypeptide chain, heavy polypeptide chain. So immediately I'm thinking, right, what does polypeptide chain mean? I know that that means protein or a sequence of amino acids. So I'm immediately painting that picture and bringing that all together. Now I've been doing this for a lot of years, so I'm able to do that very quickly, and hopefully, when you've put in enough revision, the same thing will happen as you try more of these questions. You'll start to do this process automatically without thinking about it. But I'm trying to gear you towards the tools that you'll need to know how to do this. So we can also we can see from this variable region that's kind of shaded at the end there. So once I've read all that information, the next thing I'm going to do is I'm going to put key terms down the side. So what that means is I'm looking at that question before I even kind of think to look at the set. I'm looking at it because this isn't just a ticky box or a label question. There's a little bit of extended thinking required. I'm going to have to use some key terms and some things around that. So I'm going back to what I know about antibody structure. I might be thinking about this bit here I know is the hinge region. So maybe I'll add that term in there just so I can put it in. I've had light and heavy. I can see them front there. I might talk about antigens, because I know that antibodies attach to antigens on pathogens to neutralize and agglutinate them. Those might be terms I would use. But then I'm also going to kind of think about bonding. So I know that the bonding that holds the chains together here is disulfide bridges. So I'm just giving you an example there. I mentioned a few more terms, so you could build up along the side of the question there a few terms that you're going to kind of use and interpret. So the question then says, outline how the structure of an antibody molecule is related to its function. So I've got to circle the command word there and say, right, I'm outlining. I'm not describing, I'm not explaining, I'm outlining. So that means give a general overview of all the aspects of how the structure, so how it looks, how it's made up of the antibody is related to its function, is related to its job. So I'm crystal clear on what I need to do in terms of adding that in. So what I'm going to say there is I've got 3 marks, I've got to make 3 points. I'm going to explain the 2 areas that I've been given. I'm going to talk about the constant region and the variable region because they're 2 parts of its structure. And then I'm being told also that there are these heavy chains and these light chains which are joined together. So I'm going to talk about how they're joined together and that bonding. So first of all, I'm going to say, well, the variable region binds to antigens. And the reason, antigen, let's just spell that right. The antigens are going to be different for each different pathogen because antigens vary. The antigens vary, and therefore our antibodies have to vary. So that's where the variable region term comes from. So they're going to be different, and the term we always use in biology is specific. So that might have been one of the terms that we've stuck up here. So now that I've put that in, I've talked about that particular part of the question. That's one down. I'm going to talk about two more. I'm going to talk about the constant region. So I'm going to say, all right, well, the constant region is specifically designed for binding to receptors on phagocytes. So it's constant because it's the same in all antibodies, no matter what the variation is on the variable region. So that allows binding to phagocytes, or examples of those such as neutrophils or macrophages or etc. So yeah, so that allows binding to those. So that's its structure, and that's how it's related to its function. And what holds that constant region together and the light chains together would be the hinge region. Now this isn't on the question in the stem of it, so this is something that we'd have to bring to the question prior to that. So we might say the hinge region. What the hinge region does, it gives it flexibility, so that when it is attaching, the antigen in here, here's the antigen here that it's binding to on the surface of the pathogen, it can adjust and it can move and tweak its shape so it fits in, a bit like putting your hands and your fingers into a glove. The shape of the material is flexible, it allows accurate binding. So that happens there as a result of that structure. All happens as a result of the bonding that gives us this structure, and that happens through disulfide bonds. So we'd have to remember that it's disulfide bonds between the chains. Remember that happens between cysteine residues, amino acids, where we have a sulfur in each R group of those cysteines. We get a disulfide bond, and that holds the two chains together. We're talking hydrogen bonds and ionic bonds. We're talking bonds between the amino acids within the polypeptide chain. So that wouldn't be accurate. So lots of STEM that question. That's 3 marks. So obviously we can't spend that much time in that analysis on each question. But if you're getting the point in your exam past paper practice where you're going through questions and you can't seem to get full marks and you seem to be off the mark, then maybe you've got to sort of like guide yourself through this thought process for a few and train yourself to answer questions in that fashion. So if we look at the mark scheme here, this is taken from the exam software I was using to pull the question. You can see all of those parts. Maybe it's not specifically exactly what I've said, but you can see they all relate to the points that I was talking about. So it says any 3 from the following: award mark only if the structure related is to that particular function. So what that's saying there is that if I have said there are disulfide bonds and that enables the binding to an antigen, but I've not qualified it talking about the variable region, etc., that wouldn't be specific enough. So you can see those and you can read them and have a look and just pause the video and work through it. But that's maybe how you answer a question that particular area, but this could be any topic and works for the same thing.